Welcome, Trey.
Okay, great. Thanks very much, Teresa, and thanks also to Chink and Ben and Ron and the others who invited me. It's been great so far. It's a great venue. So what I want to talk about today is a new initiative that we're trying to build critical mass in, and would love to involve anyone who's interested. It's easy to involve you because right now it's just talk, but hopefully I can convince you that it's describing what a lot of us are already doing, and furthermore that maybe a lot of what we're talking about here should be sort of focused and at least to the point where you can get some visibility for it as sort of a movement. And so far we're calling this the Cancer Cell Map Initiative, and it does involve some starter funding. It involves my collaborator Nevin Krogan at UCSF, our cancer center directors both at UCSD and UCSF, and again we're trying to sort of get some critical mass. I also found out today by reading the news that it's apparently World Cancer Day. So it's a good day to be talking about cancer. But what I want to talk about is what I think a lot of us have been talking about, which is what to do now. So later you guys can tell me what this number really is in terms of, is it 10,000 genomes we now have? Is it exomes, genomes? What's the difference? How many is it really? But I think we all agree that not only do we have a lot of genomes, there's certainly a lot more to come when it comes to cancer. Maybe hundreds of thousands of more to come in the near future if what you hear is true. So what are the challenges? As all of us, or most of us in this audience, appreciate and have been talking about at this meeting, one of the biggest challenges that becomes immediately apparent after looking at these cancer genomes, and is pointed out by all of the papers, is that most tumor mutations are rare. And that is to say, each patient is a snowflake. Their somatic alterations are completely different from patient to patient. Now, this is our version of what other people have drawn and called the long tail of mutations. Certainly Bill Hahn has talked a lot about this, and I think has made this point very effectively. And so here's a sort of a complementary take on that. So if the goal, there's different goals one might have from analyzing cancer genomes. Certainly the ultimate goal is to treat a patient properly. But in the meantime, there's been a lot of interest on, can we identify all of the cancer genes from cancer genomes? So here's why I think not only can we not, but sequence until you're blue, you're never going to be able to. And so here's how that argument goes. If we take a random patient from the TCGA, this happens to be of the 1,000 or so whole exome sequences that they've elucidated and published. This is just one randomly drawn. It's the first one that I drew. It's not carefully chosen. And we look at the somatically altered genes in that patient. There are 25 of them. This happens to be actually below the average for the cohort, which is up more towards 50. And now we plot on the y-axis here mutation frequency. And the scaling here is just versus background. So 1 would be roughly the expected mutation background of any gene chosen at random. And 50 would be 50 times that background on average over the whole genome. You now see this exponential tail where there's essentially a single gene in this patient, GATA3, that's found recurrently over the breast cancer cohort. In fact, is 50 times roughly more likely to be mutated than the average gene in the breast cancer cohort. But then very quickly, as this shows, that falls off. So now when you're looking at the 4th or 5th most frequently mutated gene in the cohort that happens to also be in this patient, now you're actually beneath what the current algorithms or statistics can distinguish as being significantly or recurrently mutated in the cohort. And in fact, a off-cited example of this is BRCA1, which was originally identified from gene linkage studies in germline mutations. In the somatic space, it actually is not identified as a significantly mutated breast cancer gene. We, of course, know it's important. And then we've actually labeled 2 other genes here, which are known cancer genes in breast cancer, and look at where they fall. The others aren't well described. So this line here is sort of our current power with the current number of genomes and methods at distinguishing what are the cancer genes from the non. And certainly it is true that as you do more sequencing, this line is going to fall. Right now it's, say, around 1, 2, 3, 3.5 times background. This will push down as low as, say, 1.5 times the background mutation rate. If you sequence, say, 100,000 more genomes, you can look at that, and we can discuss that at length. But the point is, the situation fundamentally isn't going to change, because half the genes in this patient are so close to the background mutation rate, or even below the background mutation rate. And you can think of reasons why functional cancer genes, because they say disrupt key processes in normal cells, might actually not be frequent events at all. You're never going to access this half the tail. There's just no way to do it. And so the question then is are these important cancer genes or are they not? So to look at that, what we did is go to this popular Sanger cancer gene census. You can look at the Fogelstein list, you can look at any number of lists, and the conclusions are qualitatively the same. So just bear with me on this particular list. If you look at the cancer gene census, it's about 500 genes that are, say, hand-curated, and we can argue about if the curation is liberal or conservative, but it's a gene set that's thought to be associated with cancer. And now what's interesting is if you mine back into PubMed, the first citation of that as a cancer gene for each of these genes, that's not something they've done. That's something my former student Matan Ha-Free did. You find that about 10%, just over 10%, have been in fact found through tumor genome sequencing and have been recently put in that database. But the other vast majority were found by a collection of other mostly functional approaches. So you can see here the big winner is knockout mice and other knock-in, knockout types of experiments in animals and cell lines as the way of first demonstrating a cancer gene. Many of these have indeed been rediscovered in sequences, but I think it's important to realize that most of the genes we consider cancer genes have not been found by sequencing, sequence-based enrichment kinds of approaches. So to look a little further at this, we took a collection, what we hoped was a broad collection of methods that had been proposed to analyze tumor genomes to find cancer genes. I hope most people's tools are on here, but I'm sure that some have been left off. We apologize in advance. But nonetheless, it gives you a sort of representative look at the performance currently of the methods that have been proposed or used popularly, especially to identify cancer genes. And so what no one has ever done before, all of these approaches focus on essentially precision. How good are they at recovering those known genes? And they're actually, many of them are quite good. Okay, notice we're clustering up here in terms of just reading, and we're not in most cases rerunning these algorithms. In some cases we had to. Mostly we're just looking at the supplemental table that algorithm publishes. So you can see they do quite well at finding cosmic or a Sanger center cancer genes, but quite poorly generally in recall. Even when you put them all together, you can push up recall, but of course at the expense of precision. So this is the state of affairs, and we're nowhere near, of course, where you want to be in terms of finding all cancer genes. Also, apologies to Ben Raphael here. He never proposed HotNet as a cancer gene identifier, but we just kind of came up with a trivial way in which it does identify new cancer genes, and maybe it could be made better. But just to give us a sort of feel for things. So I think a debate needs to be had not just about what you do after you find the cancer genes, but whether genome sequencing can even find the cancer genes in the first place. That's the statement I'd like to propose for later discussion. I guess point one for later discussion. So these observations for us, and I think from this conference, clearly many of you have led us to conclude that we really need further information beyond the genome sequences alone. Of course, genome sequences are required, but we think they need to be supplemented with information about other aspects of cancer cell biology, which these days are mostly captured in the so-called genetic network or molecular networks. So the point of the Cancer Cell Map Initiative would then be, like all good geneticists, we're trying to connect genotype to phenotype, ultimately to response to therapy and disease diagnosis of an individual patient. But rather than go directly from genes to phenotype, as you're essentially trying to do when you look for recurrent mutations in cancer. Let's go through a middle layer, which would summarize cancer cell biology as we know it. And our current best representation of that is a network model, although at the end of my talk I want to question that. And so the Cancer Cell Map Initiative then has sort of two prongs. One, how do we accumulate this network knowledge in the first place, and can we do it systematically, much like genome sequencing has systematically put genotypes into the public domain? And I'm not going to talk really at all about that. That's been the purvey of my collaborator, Nevin Krogan, who's using mass spectrometry-based approaches now to systematically go after some of these cancer genes. There's a multitude of other approaches one could apply that we should talk about. But given the time I have, I want to talk more about this orthogonal idea of once you have network information in the public domain that's, say, more systematic or increases in how systematic that network information is, what do you do with it and how can you use it in translation of genotype to phenotype? So what I'm going to just briefly touch on, because it's an older paper now. Last time I presented this, Ben said, God, it's a 2013 paper, he's right. So I'm just going to just refer you to the paper, but I guess the reason why I still talk about it is I'm still excited about it, I guess, and I still think it points the way to maybe some future studies into how you do this. But please do check out this paper. The basic idea is, given network knowledge, okay, you can now start to find recurrence at the level of networks as opposed to the level of genes. Certainly HotNet pointed this out earlier using network propagation. Here, essentially what we've done is we've rotated this matrix, and instead of clustering mutations in networks, the focus is to cluster patients using the propagated value. So we propagate mutations over a network such that now instead of a patient being represented by a 1 for the genes that are altered and a 0 otherwise, or some maybe functional score for the genes that are altered. But most genes not being altered, right? It's a sea of zeros and a few non-zero values. After network propagation, what you get for each patient is more of an information-rich profile, where essentially every gene has a non-zero score representing its proximity to mutations in the network. Okay, so just the raw mutations get transformed to proximity to mutations, and that's what network propagation does. That's what others like Ben have used. Certainly Roded Sharon, I think, is the first maybe to use that in bioinformatics in a paper earlier from his group. So there's nothing new algorithmically other than the way we're applying this propagation tool to transform a single patient's vector to a measure of network proximity to mutations for each gene. Yeah?
On the left-hand side, so you show a matrix. It's a patient. So the entry is so what's
yeah, so actually this is misleading. This is the actual clustergram after you've done clustering the data. This is just showing that if you try to cluster the raw alteration scores for whether a gene is mutated or not, you essentially can't cluster it. It's not robust because essentially every patient is different. You have maybe 1 or 2 features that are found recurrently, like P53, and can be used to find cluster structures, but those certainly don't provide robust clusters. After the fact, you can cluster the data quite nicely, is the idea. We're clustering patients.
So what's your observation like for your clustering? That's my question.
What's the observation?
Yes.
Yeah, so I'm getting there. So the idea is now you can take that patient by gene matrix and cluster it. You get robust clusters using bootstrapping or consensus clustering in this case. You can see this effect with a variety of networks. Maybe for discussion later, too, I would love to discuss what is the best network we should all be using for this kind of stuff. I don't think it's any of these, by the way. But I'm not sure what it is, but let's talk. But then here's some of the results. So here's cluster 1. This is the Ovarian TCGA cohort. Not that it doesn't really matter for this sort of high-level narrative, but this is that cluster 1. This was the TCGA Ovarian somatic genome or exome cohort. This is the network that supports cluster 1. So the idea here is that every patient assigned to that cluster mutates one of these genes, typically, not more than one. Sometimes it's more than one, but typically one. And it's a different gene, typically from patient to patient. That's shown here by looking at these genes versus the patients, except for Titan, which is a whole story into itself. But the other genes in this cluster are essentially getting hit once or twice in the cohort. But the important meme that you're learning is that everyone in this cluster gets hit somewhere on this page. And these clusters do, in our hands, typically correspond to survival groups, which is an important validation. So here's the survival curves that you can see that these clusters do stratify the ovarian cohort by survival. What is not published by us yet, but is encouraging, is in a more recent study of ovarian cancer genomes coming out of the ICGC. We did not relearn these networks in any way. This is all unsupervised analysis of TCGA, but now we can take that network we've discovered and use it to classify new patients, and in fact, you get the same exact stratification with patients hitting that aggressive subnetwork I just showed you, now stratifying out by poor survival. And you can, in fact, look at the difference between the TCGA cohort that was used for the discovery process of this network and the ICGC cohort that was now used to draw that second survival curve, which I guess you could call validation. This is that same network I just showed. It's just not pretty compared to that last one. It's the same connections and genes. And you can see that really there are just a few genes here, 4 of them, that are hit recurrently in both cohorts. But both cohorts are sort of just missing one another in the same network neighborhood. So it's pretty interesting. And most of these colored nodes represent just a single patient mutated in either TCGA or ICGC.
Have you assessed whether or not these mutations are believably functional?
Yeah, so that's a whole story in and of itself. So the answer is, for some ovarian is actually the outlier. Nothing we have done improves or leads us to believe, A, that these mutations are not functional, or that we could do better at finding these mutations if we pre-scored the function. So lots of people have suggested to us, including like all 4 of our reviewers back in that 2013 paper, that rather than 1s and 0s for what genes are altered or not, because we know that, in a sea of 100 mutations to exomes in a patient, maybe 6 are actually functional, right? So the theory that must be going on here, and we can show that these clusters are not found if you randomize and all of that, right? We can talk about all the controls we've done. We think we've controlled for this properly. So one would think that by applying one of these functional scoring algorithms to the 100 mutations in a patient and propagating those scores, you might do better. We actually have not found that to be robustly the case. In fact, for every cancer but ovarian, we can throw out synonymous mutations, and that doesn't have any effect. It increases the signal slightly, we find. In ovarian, it turns out there's been this argument that even synonymous mutations affect expression levels and that kind of thing, and so we actually don't even throw those out. Happy to talk about it. Let me keep going because I want to get to the second half of my talk, but it's really interesting, and I agree the jury is definitely not out. So let's chat. But yeah.
Do you want this to be a single connected component, or this is just
There is no requirement that it be, if you think about it. All we're doing is propagating that vector and clustering the result. And so the fact that it is here, so let me now admit that we don't really know how to pinpoint the right network here. What you're looking at is a heuristic. So I don't think that's a solved problem. I can tell you offline how I derived or how Matan came up with this exact set of nodes and edges, and maybe you'll agree it's reasonable, but maybe not. So let's chat. Really what you want, in thinking about this, is there's sort of a Bayesian of attraction, if I can use that language, for this subtype. And so there might even be nodes and edges or nodes that could be mutated that have not yet been observed in any patient. But nonetheless, you'd want to put in this thing because the prediction would be it's in that Bayesian of attraction. And if you were to observe a patient, say forward simulate, make up a patient and put that patient in and run her genome through, then she would have to cluster with this. And we're not there yet. There's no node. The gray nodes here are basically just connected to the colored nodes in a very trivial way. And I'm not convinced it's the right way. But approximately, we think we got it right. Not exactly. Okay, but let me, in the last 20 minutes or 10 minutes of my talk, I want to move to sort of what we've now been spending our time doing since 2013, which actually was not, you might have thought we would have made progress in advancing this method in some way or other, and we're certainly interested in doing that, and I have a postdoc working on it. But our I think, more encouraging results in the past couple of years have come from going back to model species like yeast and looking at the process of what's going here or happening here, where you aggregate mutations in networks and really try to ask whether this is the right model at all. And what I want to argue is maybe there's a better way of doing it based not on networks but hierarchies of the same information. So I want to first talk about this very talented grad student's work in my lab by Mike Kramer for how you would assemble hierarchical maps of components and functions in the cell from network knowledge, and then pass this off to Mike Yu, another very capable grad student, and show you how he's then been using these data-driven hierarchical maps to aggregate mutations in much the way I just showed you using networks. So the motivation here is that networks generally don't look like the real contents of cells. When I draw the network of, say, a Pentium chip, I can actually zoom into the Pentium chip and find the wires. When I draw a network of, say, a brain, I supposedly can find the neurons and the axons and trace those. So it actually looks like a network. These hairballs we draw for cells don't look like that. They look like this. This, in fact, is the proteasome, and this is what the proteasome, this is now in yeast. I warned you I'm going back to model species for the rest of my talk. This is what the proteasome looks like in yeast if I had shown you that in a hairball diagram. I have color-coded here the subunits of the proteasome, so you can see kind of how they lie. And the core here does kind of, in a spring-embedded layout, move to the left. And the regulatory subunits of the proteasome do move to the right. This thing is a vacuum cleaner, by the way. The tube is like that, so you're kind of looking at the side of the tube. But how do you get closer to this, or at least capture some key aspects of cell structure and function that networks do not capture? And two of those key aspects, at least, are modularity and scale. So here we see clusters of protein interactions coming together into subunits of a protein complex, but then subunits assemble with other subunits to make bigger subunits. That's, say, the red part or the blue part. And then that entire thing makes up the proteasome. I can keep going upwards in cell structure and function from here and say this is part of protein degradation and so on and so forth. So how do we capture that modularity and that scale? So around the same time of that 2013 Nature Methods paper, we had another paper which I think would prove later to really converge with or bring together both ideas. And that was this work by Janusz Dukowski and my group together with Mike Kramer. And then Kramer went on with Vineet Bafna's lab to refine the algorithm. But the idea is that a lot of this hierarchical, multi-scale, modular structure is actually present in these network hairballs if you look in the right way. To see that, you cluster the networks, just like many clustering algorithms have already been proposed to do. But as many of you know, instead of building a binary tree, which is what your standard agglomerative clustering algorithm does, you want to build a DAG. Why? Because you want the intermediate nodes of your clustering dendrogram to reflect true structure in the data as opposed to the binary tree, which is a computational convenience. And so, for instance, if these 7 proteins in the proteasome really should be brought together at once because they, in fact, are identical subunits of a larger structure, why shouldn't you just merge them all at the same time? Why force this thing into a bunch of arbitrary binary decisions, which are just going to be hard to interpret biologically later because they're meaningless? By the same token, as we know, there's pleiotropy, and so nothing constrains in the cell a protein or any module from being a member of 2 or more parent modules. And so that also has to be respected when you discover the cluster structure inherent in the network data. So then you have a DAG, a directed acyclic graph, as opposed to a binary tree. Here what you're seeing is back in yeast, we've just clustered all information that one can find in the public domain relating to yeast protein-protein interactions, co-expression interactions, and synthetic lethal interactions. You then find that this DAG resembles to a large degree the literature-curated gene ontology. So all that great work that Go has done over the years also builds a DAG, but they just decide when to create a term. So if you think about it, all the computational work with Go has been bent on not the DAG structure itself. That's assumed given. It's how you annotate new genes or gene functions into that. Here, we're actually less interested in that than the structure of the DAG itself. That said, given the literature-curated reference map of a cell, just like you can have a reference genome, this is now the reference structure of a cell, you can now align that whole thing in a one-to-one way, not Go enrichment, but a one-to-one alignment back against the DAG you've created from data. And every time that you see a match, and we can talk about how these ontology alignment algorithms work, there's many of them from computer science, one can choose. We did not write our own. That said, there's probably room for improvement. We could talk about when you find an alignment, then you can transfer any annotation Go put on that term over to your term. And equally, or maybe more interestingly, when you do not find an alignment, you might have discovered a new component in the cell. Here's what the thing looks like. Yeah?
When you do this comparison, do you have a way to take the gene ontology without network information that was aggregated?
Yes and no. So we've addressed that in a first order way, but there's a nasty second order way that I don't know how to control for. The first order way is yes, there's evidence codes, and you can use those to remove evidence due to any data that you think you've used. But I'm not sure that handles the following case. Let's say I remove this gene annotated this term because of protein interaction, I don't know, omics data. But then that gene actually interacts with another gene. I don't know. I'm not making sense, but I'm worried about second order effects here that you simply can't take out by evidence codes. But yes, we have taken out evidence codes for what it's worth. So here's what, for yeast, our data-driven ontology, which we called NeXO, looks like. And every time we label a term, it's again because we got a good alignment to Go, and so you can transfer that term annotation over. And and this is the root is here. It is a rooted tree, but it's not it's not shown top down. So there's the root, and you see very quickly this thing trifurcates into a membrane, a mitochondria, and an intracellular part, just like Go. So that maps over. You can also see that in Go, your annotation is much better for the components in the intracellular compartment than for the membrane or the mitochondrion, which is that that just doesn't get labeled. And if you look, Go has essentially very little structure beneath those parts, yet the data suggests otherwise. So again, just in passing, an interesting finding. But then to zoom in, just to kind of show you low-level detail, you'll appreciate this fractal thing is a multi-scaled structure, like any hierarchy. And so here, let's zoom into the proteasome, which is that structure I showed you before. Here I only had the hairball. Now here's what it looks like in the NeXO data-driven ontology. So it does recognize that this whole thing is these proteins are clustered together. So that aligns, it turns out, to the proteasome complex in Go. But then this thing splits into a regulatory particle and a core. That's roughly the left and right sides of this. So you might have picked that out by eye looking at the hairball. But then there's just no way. The algorithm can find further structure that there's no way, at least in this visualization, your eye can see. So the regulatory particle has a base and a lid, the core has an alpha and a beta subunit, and then you're kind of at the proteins. So that's sort of the alternative view of the same data that we think might be a lot more useful biologically than this guy. Okay, so the idea then is that maybe when you look at these hairballs of interactions, you're just too close to the data. You're looking at the raw data, sort of the raw pairwise relations that have been pulled out experimentally in various omic ways. And maybe that's sort of like, at one point people stared at these X-ray diffraction images to get the structure of DNA or structure of proteins. But now, I'm not a structural proteomicist, but my understanding is you kind of just assemble a million of these images and you press the button and you get that. So maybe we just haven't yet developed the right algorithms that, if not the full structure of a cell from omics data, maybe at least take you to an ontology. And maybe this is as far as you can push it, maybe not, but it at least makes the point that maybe the hairball is just too raw. Okay, so I basically am out of time, but I just wanted to just quickly say that we think, just to sort of pull this back to the cancer interpretation of genomes mode, we think that the way these ontologies can then be used is to process information. And this is about to come out, so luckily you can read about it instead of hearing me tell you about it. But the idea now is what we're trying to do is to percolate genotypes. Now here we're in yeast, so you typically have 1, 2, or 3 mutated genes, and the rest are blanks. But you can now percolate these down through the ontology and translate a genotype to what we're calling an ontotype, which is just instead of a set of genes that are altered, it's sort of like a set of Go terms, or in this case, NeXO terms that are altered. And you can then use those as features for prediction of phenotype. It's working really quite well. I'll be happy to talk about it offline, but let me instead get to the end of my talk and summarize. So here is sort of the overall system. You have a genotype. It's raining mutations down on your cell, but your cell is organized hierarchically. And so you can think about each component in that hierarchical map as like a funnel that catches some number of mutations, filters it through the function of that component, and then passes on that information to the next layer. And so I think this might be sort of a new and interesting way, mostly is yet to be explored, for how you might connect genotype to phenotype. Thank you.